Hello everyone, welcome to my poster presentation. Today I will be presenting on the spatiotemporal regulation of neurogenic networks by OPA and OC during Drosophila embryogenesis. Early embryonic development involves a critical transition from maternal to zygotic control. In Drosophila, this includes activation of a brain lineage markers in the anterior neurogenic domain. This second wave of gene expression is regulated by OPA and OC to conserve transcription factors, which is analogous to ZIG3 and OTX2 in vertebrates. They control genes like TOI and EMS, which are essential for a brain patterning and neuroblast specification. Understanding their regulation helps us uncover conserved mechanisms that shape early brain development across species. To study spatial gene expression, we collected staged Drosophila embryos from genetic crosses and performed FISH. We use fluorescent RNA probes like EMS, TOI, OPA, and OC to label nascent transcripts and imaged those embryos using super resolution confocal microscopy. In the result panel at the top, we show ChIP-seq data for TOI and EMS. Strong binding peaks upstream of both of the genes suggest that OPA and OC directly regulate their transcription. Below, we show in situ FISH images across different genotypes. In yellow-white, that is a wild type, and in US Lexi, that is our controls, we observed normal anterior and expression of a TOI, EMS, OPA, and OC in early as well as late embryos. However, in OPA RNA-I and OC RNA-I embryos, we saw clear loss or a reduction of a TOI and EMS expression, or we can say they are not co-regulating together. This demonstrates that both OPA and OC are required for proper activation of these genes during neurogenesis. As you can see on the right hand side of the embryo images, there is a 4.5 times zoom in images which highlights the expression patterns at the nuclei level, showing how transcription is spatially altered in knockdown conditions. And here we have shown ChIP-seq data which shows that how OPA is regulating OC. And as our next steps, we are planning to use CRISPR-Cas9 line of EMS and TOI to understand its role, as well as we are also implementing super resolution live imaging of nascent transcripts to track real-time gene expression dynamics during early neurogenesis. Ultimately, our goal is to build a detailed dynamic gene regulatory network for brain lineage specification work that could offer insights into the basis of neurodevelopmental disorders like ADHD. I would like to thank my PI, Dr. Kurumila, for her mentorship, our lab members for their support, and UTS STARS program for funding this research. Thank you.